Hello and welcome. My name is Dr. Suzanne Minton and I'm the Director of Content Strategy at Certara. This webinar is brought to you by Certara, where we accelerate medicines using biosimulation software and technology to transform traditional drug discovery and development. Our clients include more than 2,000 global biopharmaceutical companies, including leading academic institutions and key regulatory agencies across more than 62 countries. Before we begin, I'll cover a few housekeeping items. If you experience technical difficulties, please message the organizers using the chat panel. During the presentation, all participants will remain in listen-only mode. As a reminder, today's webinar is being recorded. We'll email you a link to the recording within the next few days. You'll have an opportunity to submit text questions to today's presenters by typing your questions into the questions pane of the control panel. You can send your questions in at any time during the presentation. We'll collect them and address them at the end. Today's presentation is Advancing Formulation Excellence with Simcyp Biopharmaceutics. Our speakers are special guest, Professor Jennifer Dressman, Kevin Romer, Himanshu Mishra, Dr. Nikunj Patel, Dr. David Turner, Dr. Yan Yip, and Dr. Swati Jaiswal. Professor Dressman retired as Professor of Pharmaceutical Technology at the Goethe University in 2021, and she now leads a formulation group at the Fraunhofer Institute of Translational Medicine and Pharmacology in Germany. Kevin is a Vice President at Certara and leads the sales and business efforts for Simcyp products and services. Himanshu is a Director of Business Development for Certara's Simcyp division. Nikunj is a Senior Director of PBPK Consultancy Services in the Simcyp division of Certara. David is a Senior Principal Scientist in our Simcyp division, and Yan is a Senior Scientist. Welcome to the presentation. I'll now turn it over to my colleague, Kevin, to kick us off.
Thank you, Suzanne. And thank you all for joining us. We are so excited to announce the continued expansion of our industry-leading Certara Simcyp family of products. For over 20 years, we've been developing the Simcyp simulator to support the needs of scientists and regulators throughout the drug development process, from preclinical through clinical to post-marketing. Recently, we have spawned new family offerings that focus even deeper on specialties. Last year, we announced the Simcyp Discovery for early development, and this year we are pleased to announce Simcyp Biopharmaceutics. Simcyp Biopharmaceutics is ideal for the needs of biopharmaceutic, formulation, and CMC scientists, and like the original simulator, supports them from preclinical to post-marketing. Our close relationship with biopharmaceutic, formulation, and CMC scientists guided our packaging of the powerful features and capabilities of Simcyp Biopharmaceutics, spanning in vitro mechanistic analysis with our CIVA module, through the most advanced modeling of the GI region, a variety of formulations, IV, IVC, and E, and virtual bioequivalence in lieu of trials. This base platform provides an essential and powerful tool, which we are offering at a disruptive price. To put that in perspective, the full simulator, which has enabled the approval of over 100 novel drugs and 325 label claims in lieu of clinical trials, has the perception of being more expensive than the competition. However, we are offering the Simcyp Biopharmaceutics capability at about 1/3 the price of the full simulator. At Simcyp, we provide software tools for our clients to use, and we offer tech-enabled consulting expertise to support these efforts. While this is the first year we are providing the Simcyp Biopharmaceutics tool to the market, we have a long history of using Simcyp technology in over 35 consulting projects, growing annually, to address a wide range of biopharmaceutics applications and case studies in partnership with industry and regulators. My colleague, Dr. David Turner, will delve deeper into the biopharmaceutics product product and some major case studies to illustrate its features and benefits. Before David joins us, it is my honor and privilege to introduce Professor Jennifer Dressman. It is one thing to hear why Simcyp Biopharmaceutics is important from our Certara Simcyp team. It's more important to hear a perspective from one of the premier thought leaders in the world. Professor Dressman recently delivered the opening keynote address at the Global Biopharmaceutics Modeling Workshop sponsored by the FDA. Welcome, Professor Dressman.
So I'd like to welcome everybody today to the webinar, Advancing Formulation Excellence with Simcyp Biopharmaceutics. And there's been a lot of recent interest in biopharmaceutics modeling on the part of the FDA. And in fact, they've had a couple of recent events at the FDA in conjunction with the University of Maryland, Thursday, on dissolution that was held in May 2023. And in August 2023, we had a specific workshop on PBBM, or physiologically based biopharmaceutics modeling, with over 200 participants. And it included regulatory authorities like the MHRA, Health Canada, ANVISA, and Japan. So this all underscores the interest and the need for better incorporation of the biopharmaceutics aspects into PBPK. This slide shows the philosophy behind biopharmaceutics in vitro, in vivo extrapolation, in other words, PBBM. PBBM has to be informed by in vitro experiments, some of which are shown on the lower left-hand corner of the slide. Solubility experiments, dissolution experiments, CACO2 cell and other uptake types of experiments have to be done in vitro. And so from those results, we can build mechanistic models of how a drug or a formulation is going to behave in vivo. And so we need to have good confidence in those in vitro experiments, but also in the intrinsic parameters that are generated from the mechanistic modeling prior to their use in a PBPK model. So how does all this work? Now, I've been very interested in vitro experiments like solubility and dissolution, and particularly biorelevant dissolution, for at least the last 25 years. So I'm excited by what Simcyp's offering here with their Simcyp Biopharmaceutics. So what this is essentially is the CIVA program to do the mechanistic modeling of the in vitro data, and that's fed into then a compact version of Simcyp, and that's tied to a bioequivalence module, so that in one program you can actually flow through from the mechanistic modeling of the experiments through the PBPK model into the bioequivalence module. And just to show how important it is for the mechanistic modeling, I want to use the example of solubility. And the global solubility equation is shown on this slide. And you can see at the very end, there's a term in there for drug that's bound. This could be either to a binding excipient, like a cyclodextrin, or a polymer, like HPMC. It could be to surfactant micelles, like SLS or tweens, or it could also be used to describe what happens if you have a pH modifier, like an acid, for example, to keep a weak base in solution, even when the gastric pH is elevated. And so by generating the solubility data here in a mechanistic way, you can also calculate what the dissolution is going to be in vivo. This is very important because if you have a mechanistic model to do this, you can also modify it for each and every subject in your virtual bioequivalence study, so that you know how the pH in the gut of that person, virtual person is going to affect the solubility of the compound, how their biosalt level is going to affect the solubility and therefore the dissolution, but also how the different excipients might affect the solubility and the dissolution. So I think this is really a fundamental change in the way that we work with formulations and predicting their in vivo performance. So I'm very excited about the Simcyp Biopharmaceutics tool, and I'm sure you'll hear a lot more about it today, including some really great demonstrations. Wish you lots of success in today's webinar.
All right, thank you so much to Professor Dressman. We're grateful to have her participation in this event, and I'm now going to turn it over to my colleague, David, to give a demo of Simcyp Biopharmaceutics.
Hello, everybody. This is David Turner. I am a Senior Principal Scientist at Simcyp Biopharmaceutics, and I'm here today to introduce you to some of the features of the new Simcyp Biopharmaceutics software and some of its applications. The new biopharmaceutics software forms part of the larger suite of PBPK-based software available from Simcyp Biopharmaceutics. This includes the flagship Simcyp Simulator, the Simcyp Discovery software, and the CIVA toolkit software. The CIVA toolkit is designed for the modeling of in vitro experiments to inform the PBPK modeling. This will be explained in more detail in the next session by my colleague, Dr. Yang Ni. So what is included in the Simcyp Biopharmaceutics package? Included in the package are the mechanistic biopharmaceutic modeling tools for undertaking PBBM. Also included is the physiologically based IV-IVC module. There is a virtual bioequivalence module. One of its main features is automated CQA safe space identification, which should be of great interest to many of you. Also included in the package is the CIVA 5 toolkit, which I already mentioned. And there are other tools available that come packaged with Simcyp Biopharmaceutics, including parameter estimation, sensitivity analysis, workflow tools, and many other features. Optional add-ons include additional routes of administration, and in addition to the provided healthy volunteers population, you are able to purchase separately a pediatric population. Simcyp Biopharmaceutics is fully compatible with the main Simcyp simulator. Workspaces developed within Simcyp Biopharmaceutics are exportable to the Simcyp simulator, and this streamlines the connection between groups undertaking biopharmaceutics, PBBM modeling, and clinical groups where DDI studies may be undertaken and studies with different disease and ethnic populations may be undertaken. I've listed here some of the key mechanistic features of the biopharmaceutics module. My colleague Yan Yi will show more detail of these features when she discusses CIVA after this presentation. So very briefly, we have a particle population balance model. And one of the big benefits of this is that it can handle two different solid states of the API simultaneously. You can dose drug as freeform or as salt, and we have a complex mechanistic KSP-based salt model, which includes a database of various commonly used counterions. In mechanistic terms, we have a surface pH or microenvironment pH model, which is essential for modeling salts and formulations such as co-crystals or formulations containing pH modifiers. Also included are comprehensive particle size distribution tools, using which the popular or increasingly popular product PSD method can be applied. And as you will see in the CIVA presentation, we have excipient handling tools, which can handle in a mechanistic way the impacts on solubility and dissolution of formulating with cyclodextrins, HPMC, or various micelle-forming surfactants. These mechanistic features are common to the Simcyp simulator to the CIVA toolkit, and to Simcyp Biopharmaceutics. In terms of experience of the team here at Simcyp in biopharmaceutics applications, we have listed here some of the applications that Simcyp consultancy team has worked on, where the models were used for particle size assessment and specification, setting dissolution specifications, for looking at impacts, potential impacts of scale up and post-approval changes. They have also looked at food effects, PPI effects, virtual relative bioavailability and bioequivalence. And for extended release formulations, in particular, the IVIVC, the physiologically based IVIVC tools have been used. And you can see on the right-hand side here the types of different drug substance and products that they have worked on. These include salts, amorphous solid dispersions, various prodrugs, co-crystal formulations, and these have been incorporated into various different formulations that you can see below, including immediate release tablets and capsules, solutions, various fixed dose combination, bilayer tablets, suspensions, and so on. So you can see there's quite a wide experience in this area with Simcyp consultancy. Moving to published applications of the Simcyp tools, I have listed or shown here a whole range of different publications in the peer-reviewed literature which have applied the Simcyp biopharmaceutic tools in various ways, including in some cases safe space identification. I will not go through these in great detail. I will focus on a couple of these in the next two brief case study examples. So the first example is with bempedoic acid. This was where a clinical study had already been performed with immediate release tablets, and the sponsor wanted to develop a suspension of bempedoic acid for use in pediatrics to treat hyperlipidemia. And therefore, one of the questions raised was whether a clinical bioequivalence study was required. So to try to avert this, or in order to avert this, a PBPK model was developed and verified based on clinical mass balance results. And I've listed here some of the biopharmaceutic model inputs. These include the fraction of dose pre-dissolved in the suspension, the viscosity of the suspension, which is something we can account for with the mechanistic models available. You can see the particle size differences between the suspension and original immediate release formulation. And the in vitro dissolution was modeled mechanistically using the CIVA toolkit to provide confidence in the dissolution modeling and to provide parameters for those models in the PBPK situation. So ultimately, virtual bioequivalence simulations were performed. Biopharmaceutics safe space was defined. And ultimately, a clinical study for the suspension was waived, and this was accepted in a regulatory context. So this is one quite nice application of the Simcyp biopharmaceutic tools. A second example from the literature centers upon elagolix formulations, where again, PBPK modeling using Simcyp was applied in order to waive a clinical study. So the original formulation here is the 300 mg elagolix tablet, which was subsequently reformulated as a capsule that you can see at the top there, the 300 mg capsules. Because of the formulation differences here and some differences in the dissolution, an in vivo clinical bioequivalence study was required to show bioequivalence between the capsule and the tablet. Later on, the sponsor wished to move towards a combination dosing strategy where the elagolix dose was reduced from 300 to 200 mg. And again, here they had a clinical study with the tablet, and the question to be raised here was whether an additional clinical BE study would be required for the 200 mg capsules. So in order to address this without doing the clinical BE study, a previously developed Simcyp PBPK model was developed, coupled with appropriate in vitro dissolution for these 200 mg doses, and ultimately virtual bioequivalence analyses performed. And the model demonstrated that the delays that had been seen in the in vitro dissolution of the capsule relative to the tablet had an insignificant effect on the in vivo absorption of elagolix, such that there is no clinically significant difference in plasma PK. And this, again, was accepted as an argument to avoid a bioequivalence study by the regulators. So I've mentioned a virtual bioequivalence several times already. So I'm going to provide you some details of the packaged VBE module that comes with Simcyp Biopharmaceutics. And you can see here on the left side a snapshot of our screen where a two-way crossover is to be performed. And you can see on the left side here that treatment 1 is the elagolix tablet and treatment 2 is the elagolix capsule. So the only difference between these two, what we call compound files, is the dissolution side related to the formulation. Before moving on, you can see on the right-hand side here that we have a wide variety of bioequivalence designs, including the basic common crossover study that I've selected here. And there are also various other crossovers designs available, including BALAMs, including several partial and several full replicate options. Parallel BE designs can carried out. And you can also see here that we have the CQA safe space crossover and parallel options available to you, which is where we automate the identification of safe space. One important feature to note that in relation to dose is that you can add content variability and assay percent to dose, and this can be done separately if you look at the bottom of the screen for treatment 1 and treatment 2. In terms of defining dissolution for input to these models, there are actually two ways that you can do this. You can directly input the in vitro dissolution, or you can choose to model the in vitro dissolution, which is suitable for certain formulations, particularly immediate release formulations. In the first case, when you directly input in vitro dissolution, you are assuming that this is representative of the in vivo dissolution, which is a big assumption in many cases. And also, it is quite difficult to extrapolate to between and within subject variability of dissolution with such an approach, because it's very difficult to quantify what those variabilities should be. So having taken these profiles and directly input them to the PBPK models, you can run your simulations and get your outcomes. The alternative, as I mentioned, is to model the in vitro dissolution, in which case between and within subject variability can be accounted for. So to do this, we would use the previously mentioned CIVA toolkit, and for the immediate release particulate formulations, we would apply our mechanistic model, which is a diffusion layer model. Within this framework, you can also apply the product particle size distribution approach, which is increasingly popular. Carrying out this process enables you to confirm that you are able to understand and model your in vitro dissolution in a mechanistic way. And it also provides the opportunity to estimate intrinsic parameters, which can then be input to your PBPK model. Using such a mechanistic approach means that the model is sensitive to the between and within subject variability of physiological parameters, such as pH, bile salts, fluid volumes, and various other factors. So how do we add within subject variability to the VBE module? In Simcyp, in our products, we can add within subject variability to physiological parameters. And as mentioned on the previous slide, this means we can propagate the known within subject variability of physiological parameters to the within subject variability of PK endpoints, such as Cmax and AUC. And the way we do this, or the way that you select the parameters to add this WSV, is to select a parameter such as gastric emptying time. You right-click on the box and select it for within subject variability. You do this for the range of parameters that you are interested in. And then you can see on the right side of the screen here that 6 parameters were selected, and you can specify their variability, their within subject variability, and the limits for those parameters. So this is a very user-friendly way of adding in these variabilities to these parameters. And for many of these parameters, we have done extensive meta-analyses of the literature to define what these limits or what the CVs should be and their limit. The alternative to this novel approach where we're adding this variability to within subject variability to physiological parameters is to add the WSV to PK endpoints. This is a traditional approach, And you can use the Simcyp tools to run the crossover analysis, for example, then export the data to Phoenix, where you can add WSV to the PK endpoints. A big assumption, a big caveat here is that the WSV is formulation independent. And that is a very big assumption. And of course, within Phoenix, you can perform further data analyses. The tools are quite flexible in terms of what concentrations or what sites you wish to assess concentrations for VBE analysis. Obviously, we have the typical or the common blood or plasma reference points. You are also able to reference other concentrations in other tissues. At the moment, this can be done in the GI tract luminal fluids or in the enterocytes lining the gut wall. And you can see some of the options on the left side here that you can select in this regard. This latter tool can be very important because there's increasing evidence that bioequivalence measured or assessed at the level of the blood or the plasma may not be the same at the level of the luminal fluids or the enterocytes. And I've shown you here a very interesting paper which explores some of these issues from some of my Simcyp colleagues. We also have, as I mentioned earlier, the dermal route and various different sites at which you can assess bioequivalence in terms of concentration. The main focus of my presentation today is the oral route, but we have a large and growing dermal team with a number of publications and a number of successes in terms of achieving biowaivers for dermally dosed drugs. In terms of how you set up a trial, this is the screen that can be used. Here we have selected a simple crossover trial. You can select the number of subjects you require in each trial, including their age range and the proportion of males and females. A key feature of this tool is that you're able to do trial replicates. So we're not running a single trial of in this case, 56 subjects. We will be running 10 separate trials of 56 subjects, and then looking at each trial separately in terms of the outputs. And I show an example here where 10 replicate runs were defined. This is a test reference ratio with 90% confidence intervals for AUC. And you can see from this example that when we take the confidence intervals to account, you can see that bioequivalence is not obtained. In this particular case, only one of these trials can be considered bioequivalence between the test and reference. So typically we are looking for, in terms of power, 80% of cases to be bioequivalence when we run multiple trials. So clearly in this case, we would reject the hypothesis that the two formulations being compared are bioequivalent. Within the mechanistic models, we are also able to account for excipient effects. These excipients can be cyclodextrin or, for example, HPMC, as I mentioned earlier, and they may also be micelle-forming surfactants, such as tween or SLS, for example. So using this type of dosing screen, we can specify how the excipient is added to the formulation, whether it's part of the solid dosage form, whether it's in solution. We can specify the dose, in this case, of excipient, and various other physical chemical properties. Then once we have characterized the binding of the API to the excipient using binding constants or micelle partitioning tools, we can then start to consider the impact of these excipients on solubility, on dissolution rate, and on free concentrations, driving the permeation of the drug through the gut wall. And one application or potential application of this excipient model is, for example, with cyclodextrins that I've shown here, where you may wish to assess whether a test formulation with 10 milligrams of cyclodextrin instead of 40 are bioequivalent to each other. Using these tools, and provided you have calculated or estimated the binding constants of the API for the cyclodextrin, you can use the VBE tools to make an assessment of whether these two formulations or different doses of excipient are expected to be bioequivalent. We also have permeability modifier tool here, where the excipient may be added to enhance permeability. So with the appropriate in vitro information, we were able to model this using these tools. In this example, I want to show you an application of the Simcyp dual solid state tools. Tacrolimus has been formulated in various generic drug products in a fully amorphous form. But particularly for one of these drug products, during storage under humid conditions, crystallization of the amorphous material can occur. So one question that may arise is, what is the impact on bioavailability and PK parameters of these different extents of crystallinity? And the ultimate question is, at what point does the formulation with the crystalline material become bioinequivalent to the fully amorphous formulation? So to look at this, we can use the Simcyp dual solid state tools, where you are able to specify the proportions of amorphous and crystalline material in the dosage form. So in the snapshot at the bottom of the screen, you can see two different solid states. Solid state 1 is the amorphous state with its relatively high solubility, and solid state 2 is the crystalline state. And you can see we've specified that 30% of the dosage form is amorphous and 70% is crystalline. And if we can apply sensitivity analysis to this and look at the effect of different extents of crystallinity, and in the above profile, you can see the impact or the predicted impact of these different degrees of crystallinity on the exposure in the blood. So you can see, as you might expect, the more highly soluble amorphous material gives you high Cmax and higher exposure, whereas the material with the highest extent of crystallinity gives you the lowest Cmax and the lowest extent of exposure. In terms of assessing bioequivalence or lack of bioequivalence to the fully amorphous formulation, we applied a two-way crossover exercise using 50 healthy volunteer subjects. And we did this for 5 different trials. So each of these trials contains 50 different subjects. And then we repeated the VBE analysis in each of these cases in order to gain some idea of the power of these trials. And in these simulations, we're at the top here, we're comparing the test reference ratio in terms of Cmax. So test is the formulation with the crystalline material and the reference is a fully amorphous material. And you can see where the formulation becomes bioinequivalent or lack of equivalence occurs where the material is more than approximately 20% crystalline material. And this varies a bit across the trials, but is reasonably consistent. For AUC, for exposure, where this lack of bioequivalence occurs is at a much higher degree of crystallinity. And this is hypothesized to be because the crystalline material is poorly soluble, it dissolves more slowly, and it tends to be absorbed in the distal small intestine. And this drug is highly metabolized by CYP3A4 in the gut wall, and the extent of this metabolism reduces as for material absorbed in the distal small intestine, and therefore FG increases. So I think this is a very interesting example. And finally, I wanted to highlight the SALT model and how it can be used for salt form screening and selection to mitigate gastric pH DDI. So we have a publication where the model was applied, and we also have an online presentation of the modeling. But essentially, famotidine, an acid-reducing agent, significantly reduced the bioavailability of a BCS class 2 low pKa free base. And therefore, various salt forms were investigated to try and mitigate this effect. And the PBPK model that was developed predicted that the sulfate salt may attenuate the gastric pH DDI. So this modeling exploited the mechanistic salt model that we have, also the dual solid state modeling, where in this case the dual solid states are the salt versus the free form of the base. And essential component of this model were the self-buffering effects of the salt on the dissolution, the generation of supersaturation without media change, simply through the salt dissolution, and subsequent precipitation of some of the drug as the free form. So we think this is a very nice exemplification of the salt models and the microenvironment pH models. Thank you for watching.
Great. Thank you so much, David, for that demonstration. And now we'll be turning it over to our colleague Yan to give a demo on our Simcyp module, or CIVA module.
Hi everyone, I'm going to talk about CIVA, which stands for Simcyp In Vitro Analysis Toolkit. This is a highly recommended standalone companion software for the Simcyp simulator and is included in the Simcyp Biopharm package. CIVA is a tool for the modeling of in vitro experiments. In the context of biopharmaceutics in vitro in vivo extrapolation, this facilitates PBPK or PBBM modeling by allowing users to estimate or confirm intrinsic parameters prior to their input into a PBPK model within the main Simcyp simulator. This reduces the need for parameter estimation within the PBPK model, which is often associated with parameter identifiability issues, as well as allows for model extrapolation to different subjects and different populations, healthy or otherwise. In turn, PBPK modeling can be used in a feedback loop to inform in vitro experimental design. CIVA has the capability to model a wide range of in vitro experiments. The main module relevant for biopharmaceutics, however, is Module 2, where users can model solubility and dissolution experiments for their API of interest. For example, and in brief, users can estimate or confirm key solubility input parameters by modeling API solubility in buffers at various pH, in biorelevant media containing different concentrations of bile salts, and in solutions of binding excipients such as cyclodextrins. Users can also estimate or confirm key dissolution input parameters by modeling common dissolution experiments such as the USP2, the USP4, or the microdis apparatus, or setups designed to more closely mimic dissolution conditions in vivo, such as the serial dilution, two-phase, and transfer models. The version of CIVA available with the Simcyp Biopharm package is version 5, which contains a host of new features. Previous users of CIVA will immediately notice a substantial screen redesign to match that of the main Simcyp simulator. The packaging is not the only upgrade in version 5, as we have also added many new models to bring CIVA in line with Simcyp version 22, and I will elaborate more on these technical updates later. Parameter estimation algorithms, which are central to CIVA, have also been improved in line with Simcyp version 22. And a small but useful update that is worth mentioning here is the addition of a simulate only button, which has a different function than the fitting or parameter estimation button. So this simulate only button allows users to quickly simulate the outcome of their model inputs as is without parameter estimation. Last but not least, on the non-technical updates, for the solubility and dissolution modules, we have added the ability to dynamically output results on screen in the form of tables and plots. Here, following parameter estimation or simulation, users can click on the output section to dynamically view the results. Users can select one or more experiments of interest and view the results on screen in the form of tabulated and graphical outputs. The results are grouped according to solid-state dependent time profiles such as solubility, diffusion coefficient, and particle surface pH, and non-solid-state dependent time profiles such as dissolved drug mass and drug concentrations. In addition, there's also mass balance and particle size distribution information to explore. The results in the output section can be directly copied out of CIVA by right-clicking the upper left corner of the table or by right-clicking on individual figures. Now onto the technical updates for CIVA 5. Shown in this slide is a summary of new features for the solubility and dissolution modules, for which I will demonstrate briefly. For the solubility module, the first update is the addition of a new excipient tab that allows users to model the impact of a binding excipient on drug solubility, such as cyclodextrins and polymers like HPMC. Here, users can input binding constants of the neutral and ionized drug according to the observed API to excipient stoichiometry. The second update in the solubility module is the ability to consider the API in its free or salt form. Previous versions of CIVA only had the capability to consider API in the free form. Related to this new option is the capability to model salt-limited solubility using the solubility product or KSP model. Here, users can input or estimate drug salt solubility at pH max for a counterion, which allows for the back calculation of a KSP value and the determination of salt-limited solubility based on 16 equations. This mechanistic way of calculating salt-limited solubility is applicable to both the free form of an ionizable drug, such as acids, bases, or ampholytes, as well as a salt form of a drug. This option also gives users the ability to model the effect of common ions present in the solubility media on API solubility. Now for the dissolution module technical updates. The most significant new feature here is the addition of the particle population balance PPB model to handle drug dissolution from fine particles. Compared to the previously available mass balance only model, the PPB model is more mechanistic in terms of its ability to track particle counts while maintaining mass balance, and therefore is our recommended model to use. One of the major advantages of the PPB model is its ability to model two solid states. For example, when the crystalline and the amorphous forms are both present in a dosage form, when two crystal polymorphs are present in a dosage form, or when the salt and free forms of the API are present in the dosage form. Dual solid states handling is not only limited to dosage form specification, but can also be applied to model drug precipitation into a solid state different than what was dosed. Once two solid states modeling is activated, users can specify the fraction of each solid state within the dose and whether the drug is present in the free or salt form. In the input fields for solid state specific parameters such as particle size distribution, aqueous phase solubility, as well as supersaturation and precipitation, users will find two active tabs corresponding to solid state 1 and solid state 2 to input different information for the solid states so that CIVA can model drug dissolution, supersaturation, and precipitation accordingly. In the aqueous phase solubility tab, and as per the solubility updates, the fourth addition to the dissolution module is the ability to model salt-limited solubility using the new KSP model. In combination with the mechanistic surface pH model, CIVA 5 now has the capability to mechanistically model the dissolution of API salt forms, including their potential to generate drug supersaturation. This is achieved by considering the KSP limited solubility, the self-buffering effect of salts on particle surface pH, and the consideration of common ions present in the dissolution media. This is a great advantage of the CIVA toolkit, since drug products formulated as salts constitute a major portion of the top 200 prescribed drugs in the United States. It is worth pointing out here, however, that the benefits of selecting the KSP and the mechanistic surface pH models are not only applicable to salt forms of an API, but also to the dissolution modeling of free ionizable compounds such as acids, bases, and ampholytes. The sixth and the last item on the dissolution technical features is the updating of the polydispersed particle size distribution tools to be in line with SimCip version 22. In brief, users can now input cumulative density information for their dosage form, for example, D10, D50, and D90 information, and fit for a particle size distribution using one of our available functions. Users may also opt to input particle size information using the discrete option, where particle radius can be manually input, or in the event of a large number of particle size bins, imported via a CSV file from an instrument such as the mastersizer. So that sums up the presentation on CIVA 5. I hope you found it useful, and thank you for your attention.
Great, thank you so much to my colleague Jan, and I'll now be passing it over to my colleague Kevin.
Thank you again. And what we're showing here on the screen, we've shared a lot of very technical and scientific information with you, but a lot of it comes down to your ability to work with the product and get comfortable with it as it would serve your particular applications. So what we've provided here is a QR code that takes you to our SimCip Biopharmaceutics landing page. And we can follow up with you in a variety of different ways. We can arrange a meeting, discuss your specific use cases and user needs. We can demonstrate how it could be applied to those needs. We can offer you a free evaluation license that we'll be hosting for you so that you can get started with it immediately. It's AWS hosting. And we can also provide you a proposal for licensing the product that scales to your needs, and that could fit with your budget for the remainder of the year. In addition, at the end of this webinar, we will be providing a survey where we hope that you'll be able to provide us your feedback that'll help us better understand your needs. So I think it's time for us now to move into the Q&A portion, Dr. Minton.
Thanks, Kevin. Yep, looks like we've got a couple questions on here for our colleagues. Looks like there's a couple for David. David, someone wanted to know, in addition to mentioned features for SimCip Biopharmaceutics, can we do gender effect evaluation with this tool?
Thank you, Suzanne. Yes, you can. We have various features associated with disposition, which have gender differences. For example, plasma protein binding concentrations. In particular, in relation to biopharmaceutics, the colon residence time of females is much longer than males, and that information has been collected from the literature and is stored in the system. And there are various other factors. Of course, we generate realistic females in terms of body weight and height and so on, and that has knock-on effects to liver sizes and hence liver metabolism. So from those aspects, we are able to deal with gender effects.
Great, thank you. Kevin, someone wanted to know what kind of training and support you can offer for these products.
Well, I'm going to actually ask my colleague Himanshu if you would respond to that. And let's make sure everybody's on camera. David, I think you want to join us on camera too. And Swati.
Thank you, Kevin. So in terms of the training and support, we run a lot of workshops in person and online throughout the year. So we provide specialized training around that. In addition to that, obviously, when you get started with SimCip Biopharmaceutics, we'll provide orientation and kind of setup and tech support help. Along with that, as a license holder, you do get complimentary workshop places and some consulting or strategic advisory support from our consulting experts. So there's a lot of benefits and training and support that we provide for our license holders.
Great, thanks so much, Himanshu. Got a question. This might be for David or Swati. Can you tell us a little bit more about the automated CQA safe space analysis? Will this be available in SimCip Simulator version 23?
Shall I take that one? Yes, this will be available in SimCip version 23 and in the new SimCip Biopharmaceutics tool. Essentially, what it does is obviously automate identifying the boundaries of CQA safe space, and you can do this in different ways. You can specify, you can scan through different empirical functions, or scan through different particle sizes or particle size distributions, and the VBE tool will automatically calculate the equivalent statistics and then identify the boundaries of the safe space in the outputs. And that's it in a nutshell.
Great, thanks so much, David. Someone wants to know, where can I find more information on regulatory accepted case studies that use SimCip Biopharmaceutics technology?
I'll go ahead and handle that, Suzanne. So if you go to our landing page, we have a white paper titled Advancing Biopharmaceutics Drug Formulation Using Biopharmaceutics Technology. And you'll see how many different projects, many different case studies have led to approvals or biowaivers. It can be easily, again, accessed using the QR code that we had before on the screen. And by the same token, we are continuing to provide that information in new formats. And we'll actually have an electronic version of that will be allow you to interact with it, and we'll show you much more graphical updates on that information coming out very soon.
Great, fantastic. Thanks, Kevin. Looks like we've got one more question from our audience before we wrap up. It's about CIVA. Someone wants to know, what inputs are required for CIVA 5 dissolution modeling?
I'll take that one. Yeah, so mechanistic modeling of dissolution in CIVA is for particular immediate release formulations. So we need, or the user will need, solubility information. So Henderson-Hasselbalch solubility, biorelevant solubility if your dissolution has been performed in FACID or FECID, for example. And because we're dealing with particular formulations, of course, we need to consider particle size. So you can input that into the system and re-estimate the particle size to give apparent particle size if you wish to carry out the product particle size distribution method. We are also able to handle the hydrodynamics of the in vitro systems and factor out the high rates of stirring that you can use in your in vitro system using a fluid dynamics approach. So this actually factors out the difference between in vitro and in vivo dissolution and allows us to map the dissolution more effectively to in vivo, even though your in vitro dissolution may be done under hydrodynamic conditions very different to in vivo. You can also account for excipient effects in the model as hinted or suggested by the CIVA demonstration. So those, the excipient can affect obviously the ultimate solubility of the drug and also the solubilization driving the dissolution. So those are the main factors that you can or need to consider.
Yep. Okay. Fantastic. Thanks so much, David, and thank you to the whole team for this excellent presentation. I'd also like to thank our audience for their attention and their great questions. As Kevin said earlier, we have a survey that will be launching at the end of the webinar, so take it and let us know how we did. We have upcoming webinars that are coming up that you won't want to miss. You can register for all of them on the Certara website. And on behalf of Certara, I'd like to thank you for attending this presentation. Have a great day, and we'll see you next time.
Thank you.
Thank you.